Good day everybody. Today I am going to discuss about the Ames test. Now to understand the purpose of the Ames test, we have to know the spontaneous mutation, mutagens and carcinogen. Now spontaneous mutation are the modification of the genetic materials like DNA, and it's occurred due to the error in natural biological process, like for example replication. Whereas on the other hand, mutagens like chemical mutagens that are also modify the genetic material like DNA, but at a frequency which is much more greater than the spontaneous mutation. On the other hand, carcinogens are those substances that are capable of causing cancer. It has been observed that many substances that are mutagenic to bacteria are also carcinogenic to higher animals. And research indicates that approximately 90% of the chemicals proved to be carcinogens are mutagens. They cause cancer by inducing mutation in the somatic cells. Now, advancement of industrial progression, development of new products, generation of several byproducts from these industrial developments creates several pollutants. And some of these pollution may be a mutagens, may be a carcinogen. So it is essential to determine their potential genetic hazards. Now, mammalian cell structure and human enzymatic pathways always differ from those in prokaryotic bacteria. But the basic chemical nature of DNA is common to all organisms. So this permits us to use a bacterial test system for the rapid detection of possible mutagens and therefore possible carcinogens. So this Ames test is a rapid, inexpensive means of using specific bacteria to evaluate the mutagenic properties of potential carcinogens. Now, after the objectives, there next is the requirements. Now, first one is the test organism, that is histidine negative auxotropic strain of Salmonella typhimurium. Now, we have to know the wild type bacteria. Wild type bacteria are called the prototrophs, and the mutants lacking the particular characteristic are known as the auxotroph. Now, what is auxotroph? Auxotroph is a bacterial mutant that requires one or more growth factors that the wild type or prototype can synthesize. Or in other words, we can say auxotroph fail to catabolize certain organic substances, or they require growth factor for their growth. Now, this histidine auxotropic strain of Salmonella typhimurium, that word means it is a mutant strain that failed to catabolize histidine and thus required histidine as the growth factor for their growth. Now, the second one is this is the second one, this is the first one. This is the second one is the minimal agarplex. Now, what is the minimal media? Minimal media contain the minimum nutrients that is required for the bacterial growth. This type of media, this minimal media, mainly used for the isolation of wild type bacterial cells. Thus, histidine auxotropic strain of Salmonella typhimurium will not grow on a medium that is deficient in histidine, that is in the minimal media, unless a back mutation to histidine positive has occurred. Now, third one is the liver homogenate, or particularly rat liver homogenate is used for Ames test. Now, when a mutagen or non-mutagen enter into our body, it is modified by the enzymatic reaction of the body. And by this enzymatic modification, a non-mutagens become mutagen, or a mutagen become more potent mutagen or become non-mutagen. And this enzymatic modification mostly occur in the liver. So, in case of a Ames test, it generally requires the addition of a liver homogenate, which serve as a source of activating enzymes that make this bacterial system more and more comparable to a mammalian test system. And the fourth one is the traces of histidine. Traces of histidine. Traces of histidine. Now, this trace of histidines allows the bacteria to undergo the several cell division that is necessary for the mutation to occur. So, this is the experiment section. Here a tube containing molten agar that contain the histidine auxotrophic salmonella typhimurium and rat liver homogenate and also trace amount of histidine are mixed together by rotation and then poured onto a minimal agar plates and allowed to solidify. After the solidification, a disc impregnated with the test chemical is then placed at the center of the test plates, and in control, sterile water impregnated disc may use. And after 24 hours at 30 degrees 7 degrees centigrade, we incubated those plates, and after the incubation, the each colonies both in the control plates and the test plates represents a histidine negative to histidine positive revertates. Control plate contain colony that arise may due to the spontaneous mutation, and if the chemical in the test plates is a mutagen, then test plates contain more number of colonies than the control plates. This is because chemical mutagen diffuse and cause mutation in the bacteria and thus produce histidine negative to histidine positive revertates. Strong mutagens gives more colony than the weak mutagens. So, this by this Ames test, we can also measure the mutagenicity of a substance and simultaneously determine its carcinogenic property. Because as we said earlier that 90% of the mutagens are carcinogens. The unmutated auxotroph present in both the control and test plates may appear as a faint growth but do not develop into full size colony because there is a rapid exhaustion of the histidines. So this is all about the Ames test. Thank you for listening.